Now I'm going to talk about translation, and I'm going to talk about four main points. I'm going to give you a very small introduction about translation. Then I will talk about these organelles. Is the organelle where translation takes place? And then I will move into the how these polypeptides come together and attach each other. And finally, I will briefly talk about the stages and I will show you videos during these four steps so you can understand in more detail what is happening. So let's start with the first one. And again, I recommend you to watch these videos to understand this section in much more detail. Now I'm going to talk about the molecular components of translation. And to do that, I'm going to talk about two main type of RNA, the messenger RNA and the transfer RNA. Why? Because a cell translates the messenger RNA using the help of the transfer RNA. And how it helps? So let me show you. The first thing that you need to remember to understand how the transfer RNA help the translation is first to know that they are not identical, because here at the end each transfer RNA will have an amino acid. And here, so they will have 20 different type of transfer RNAs. And here they have another important structure that is a triplet. And yes, you have here before the word triplet in terms of codon, but this will be called an anticodon. So the anticodon will be located in the transfer RNA, while the codon will be located in the messenger RNA. So the anticodon will attach to the codon. So they will be complementary, and this will be located in the tRNA, and this one will be located in the messenger RNA. So let's recap all the things that we said today. So I told you that the cells translate the messenger RNA into a protein, and how they do it with the help of a transfer RNA. Then I told you that the molecules of transfer RNA are not identical, and I said to you that there are two main differences in the RNAs. That is that they have an amino acid on one end. So here, for example, you can see a diagram of a tRNA, and on the end they will have an amino acid, and at the other end they will have this structure that is called an anticodon on the other end, and the anticodon base pairs with a complementary codon on the messenger RNA. So here you will have a messenger RNA that will attach. Now I'm going to talk about the structure and the function of the transfer RNA. And to do that, I'm going to use this two-dimensional structure. And just to remember, here is the region where the amino acid is going to attach, and here you can find the anticodon. And if you think about the tRNA, the tRNA molecule is a single RNA strand. So although you can see here hydrogen bonds forming, and it seems that there are two strands, it's actually one strand that it folds. So it allowed them to form hydrogen bonds, and it is a relatively small molecule. It's formed by 80 nucleotide longs. And another important fact of the structure of the tRNA is that they have this clover leaf structure. So let's recap all the things that we have said. First, I told you that the tRNA molecule consists of a single RNA strand that is only 80 nucleotides long. I told you if we flatten it into one plane, it reveals its base pairing and a tRNA molecule look like a clover leaf. In the previous slide, I showed you the 2 dimension of the tRNA. Now I want that you see it how it actually look, because it's a 3-dimensional structure. And why it forms a 3-dimensional structure? Because it has these hydrogen bonds, and these hydrogen bonds twist and fold the tRNA in a 3-dimensional structure that it shows a shape of an inverted L in this case. So let's recap. And as I said, because of the hydrogen bonds, the tRNA actually twists and fall into a 3-dimensional molecule that roughly has an L-shaped structure. Now let's talk about the 2 steps required to have an accurate translation. And here, if you see, you have here the transfer RNA located in the start codon. And the first step to have that, you will need to have an amino acid and your tRNA. And how they got together? Because they have an enzyme that spends ATP to put this product here and the tRNA here. And as a result, you can find that these two are together. So this is the first step. So a correct match between the tRNA and an amino acid, and it's done by this enzyme that you see here. The second step to have an accurate translation is to have a correct match between the codon and anticodon. So as you can see in this diagram, here you have the anticodon and here you have the codon, and they have to match perfectly to have an accurate translation.